In this video, we will study about how transcription occurs. In first video, we have studied what is transcription, which enzymes are required for the transcription. Now, in this video, we will study exact mechanism of transcription. Now, what is transcription? As we have already discussed, it is the formation of RNA from the DNA. Now, link of the first video will be given in the description box. So, what are the various stages of transcription? Now, first is when the process start, this is called as initiation. Now, during initiation of transcription, there is activation of the ribonucleotides. Now, what are ribonucleotides? They are building blocks of RNA, also called as ribose nucleic acid. Ribose is sugar, which is involved in the formation of RNA. So, we can see here, now this is the nitrogenous base, this is the ribose sugar, and this is the phosphoric acid. Now, we have taken here nitrogenous base guanosine. We can also take cytosine, we can also take uridine, we can also take adenine. So, these are nitrogenous bases which are going to bind to the sugar and to the phosphoric acid. So, these are called as ribonucleotides. So, when one phosphorus is attached, this is monophosphate, and when three phosphorus are attached, this is called as triphosphate. So, ribonucleotides, they are required for the RNA transcription. So, first step is their activation because this RNA transcription starts in the nucleus. So, in the nucleus, these ribonucleotides will be activated. So, this is called as activation of ribonucleotides. Ribonucleotides, as we have discussed, made up of sugar, phosphoric acid, and nitrogenous base. Now, there are four types of ribonucleotides. If nitrogenous base is guanosine, so this is guanosine monophosphate. If it is uridine, so this is uridine monophosphate. AMP, adenosine monophosphate, and cytidine monophosphate. Now, these are its short abbreviated form: GMP, UMP, AMP, and CMP. Now, when they have to be activated, then two phosphorus are added with the help of enzyme phosphorylase plus energy. Ribonucleotides are activated and two phosphoric acids are being added, like GMP will be converted to GTP, mean guanosine monophosphate will be converted to guanosine triphosphate. Similarly, uridine monophosphate will be converted to uridine triphosphate. AMP monophosphate will be converted to adenosine triphosphate, that is ATP. CMP will be converted to CTP, that is cytidine triphosphate. Now, this is how we'll take one example of guanosine. This is ribose sugar. Now, here it was one phosphoric acid. Now, we can see here three phosphoric acid, they are attached. Similarly, if nitrogenous base is uridine, so again this will be uridine triphosphate. If it is adenosine, this will be adenosine monophosphate. If it is cytidine, this will be cytidine monophosphate. Now, as we discussed in the first video, when RNA transcription is going to occur from the DNA. Now, this is the double stranded DNA running from 5' to 3' direction, and second strand will be from 3' to 5' direction. One is template strand, another is non-template strand. So, template strand will give rise to RNA. So, DNA is coiled like this. So, part from where RNA has to be produced, supposing this region is going to form RNA, then there is unwinding of DNA, that is uncoiling of DNA. So, we can see here, this is the point where uncoiling has occurred. So, this is the segment of DNA which is going to form the RNA. So, this will get de-repressed, and this strand which is forming RNA, this is called as template strand. This is unwinding or uncoiling of DNA. Now, when it has unwinded, now these are the, say, these are, we can say such bases are present, any kind of bases may be present, GCA, UCGA, UCA. So, opposite to them, now ribonucleotides will be added. Now, because first they have been activated, mean 2 phosphoric acid have been added. Now when base pairing has to start, that is, if opposite to G, C will be there, C opposite G will be there, A opposite U will be there, U opposite A will be there, C A opposite T will be there, right? No, T is not present in the RNA, U will be produced. So when base pairing will occur, then this triphosphate form will be converted to monophosphate form. First they were activated, they were converted to triphosphate form. Now this triphosphate form, with the help of enzyme pyrophosphatase, will be converted to ribonucleotide plus 2 phosphoric acid will be removed. Now these 2 phosphoric acid will be further, this bond will be hydrolyzed and will get inorganic phosphate plus energy. Now uridine triphosphate will be converted to uridine monophosphate. Guanosine triphosphate will be converted to guanosine monophosphate. Cytidine triphosphate will be converted to CMP. Adenosine triphosphate will be converted to adenosine monophosphate. Now if we have this strand of DNA, supposing here it is G, now opposite C, here it is C, opposite G, here it is A, opposite U. So now these bases will be added in a monophosphate form. Now this is also called as base pairing, or we can say RNA chain formation. So here RNA chain formation will start at this end. Opposite bases will be added. Now next step will be elongation, right? When bases are being added in the monophosphate form, like we can see here in this is the DNA, which is double strand, right? So this is a non-coding DNA and this is the template DNA. Now this has been shown that RNA polymerase is binding to it, right? So opposite to A, U will be added, opposite to C, G will be added, like this, opposite to T, A will be added. So this is the newly formed RNA which is being produced. So this formation of RNA chain is called as chain elongation. Now we can again see this is in this diagram. So these are the transcription factors, promoter region, and this is the unwinding of DNA. Now this is the initiation site of the transcription. So here RNA polymerase will first bind, this will keep on moving, and this RNA transcript is being produced. So this is the gene, blue colored gene, which is forming the RNA. Now what happens during formation of RNA? Now we'll study in detail. Now this is the double stranded DNA. This I have shown the bases which are complementary to each other. So this is 5' to 3' direction. Now this strand is from 3' to 5' direction. We know one is the template strand which is helping in the formation of RNA. Then we require RNA polymerase enzyme. RNA polymerase is made up of 5 kind of polypeptide chain, 2 alpha, beta, beta prime and sigma factor is associated with it. First they are dissociated, then they will combine to form this RNA polymerase during formation of RNA. Now this structure of RNA polymerase we have discussed in the first video of transcription. Now link will be shared in the subscription box. Now we can see this is the RNA polymerase which is binding to the initiation site on the DNA from where this RNA formation has to be started. Now then sigma factor will dissociate from it when RNA chain formation has started. Now this is unwinding of DNA. We have already discussed how unwinding of DNA take place, and opposite to bases will be added, and this is the formation of the RNA chain. Now this is elongation, means this is being elongated. RNA polymerase will keep on moving on the DNA strand and RNA chain will be produced, and this is called as RNA chain elongation. So whenever supposing this is the DNA, this part of the chain is going to form RNA. Now its synthesis has to be stopped over here, so this will be called as terminator region. and we can see here this is the terminator region. For that, rho factor is required for termination of the RNA. Supposing RNA has been produced on this gene, so this will be stopped over here. This is being produced opposite to this strand, and this will stop over here, right? So this is called as RNA chain termination, right? So here rho factor will be there. So when RNA transcript has been produced, now here its formation will be terminated or it will be stopped. So we can also see in this figure, now this is unwinded part of the DNA, and this is the sequence. Opposite to that, this RNA transcript has been produced. Now if template strand is from 3' to 5', now this RNA transcript is from 5' to 3'. So this is also called as newly formed RNA transcript. This is also called as hnRNA, or we can also call it as primary transcript. This is all about mechanism of transcription. Now, how this happen in case of eukaryotes, we'll study in detail in the next video. Thank you for watching my video. If you like my video, please like, share and subscribe.